So I'm going to talk about some of the design issues from a statistician's point of view for developing trial designs with rare diseases. So that's the thrust of this talk. I'm sure we'll have some other things to talk about as we go through. So we'll talk about some key components of design. Some of these cross whether they're rare diseases or not, but we'll talk specifically about some design issues for rare diseases, design challenges associated with rare diseases, and then some examples of designs that we can think about. So in general, when we're thinking about designing trials, of course those trials should answer a really relevant and important question. We're spending a lot of time and effort, usually money and person time, and we want to make sure that all of these trials are designed in a rigorous way. So we have lots of different methodologies that we can think about to answer those questions. And then, of course, we want to minimize ethical concerns and risks to patients. Let's see, I can't read that one, gosh, okay. So we know that in rare diseases, lots of times we're limited in the terms of how many patients we can recruit, especially at any one site. And then the scope of that trial. Sometimes these are diseases that are hard to recruit for or involve special considerations in terms of testing or whatnot. So we have to think about what are the variables involved in considering recruitment of those patients, outcomes on these patients, because perhaps they're a little more diverse. When we have more patients available in more prevalent diseases, those patients create more variability in what we're measuring. So that is something that we take into account when we're thinking about the estimates we might get from the patients in the end. We may, because we're limited in the terms of how many patients we recruit, only be able to detect some kind of effect that's larger than what we would typically consider of a clinical threshold. So we might need to consider something a little bit beyond what more prevalent diseases would expect to be designed for in terms of clinical effect size. Okay. I might skip through some of this, but I'm sure they'll have time for questions. So things that we might modify. So we know that when we're designing trials, we think about errors involved, the types of errors that are involved in the decisions that we make. So we talk about type 1 error, type 2 error. So those are related to the alpha level and the power of that study for that statistical test in a very traditional frequentist domain. So we can modify those thresholds. Typically, we're thinking about a 5% type 1 error rate and perhaps a 10% type 2 error rate associated with 90% power. We can think about expanding those to use 80% power, maybe a one-sided test versus a two-sided test. This really depends on the phase of research and the limitations involved in what we're studying. So those are really simple things that we can consider. Typically, we typically have a one-sided hypothesis that we're testing. So we can consider doing a one-sided test, and that translates into a reduction in the number of patients we ultimately need to recruit. We may, as I've said, we may consider what the minimum effect is that we want to detect and kind of expand that target. So as an example, so if we're thinking about in terms of hazard ratios where we're looking at a time to an event endpoint or something for a continuous endpoint, where we're thinking about designing a study in terms of standard deviation units, we may typically look at something being clinically relevant as a 1.5 hazard ratio, 50% increase in that risk. As you can see, that requires a large number of events to be observed during that study period. If we think, well, you know what, a 2-fold increase in that risk is still really relevant to what we're studying, or a 2.5-fold increase, we still want to know what those effects are. These sample size requirements are not kind of a linear function of the effect size. So with some kind of expanded idea about what we think is important, we can kind of modify the required sample size there. Okay, so some other things we can consider. Lots of times there's more than one treatment we want to study at the same time. Sometimes those treatments are provided together. Sometimes they're provided alone. So one of the things that we can do when treatments are given together is we can consider what's called a factorial design, where we cross the two treatments, and then we can kind of use the information from the numbers of patients in those. Say we have a two-group comparison and a two-level treatment and a two-level treatment, just two different treatments. We would then have 4 groups. But we can borrow information from all of those treated on one or the other. That includes the patients that are treated on both when we do the analysis. So that provides a more efficient use of all the patients under study. We could then look at the differences between those who get single agents versus the combination of agents. So this is something we can just think about considering in our pool of resources in designing these trials. The other thing we can think about, and we don't do this enough, is doing trials with multiple arms. So lots of times you'll see this, and I know there's lots of complications in doing this all at once. We can do one bigger trial with multiple arms rather than singular trials in succession. So we have 3 agents. We want to study all the agents or all of the regimens, whether it's how we give the treatment or the actual dose level. We can consider combining those scenarios where we look at multiple different arms. And this, again, provides some efficiency in terms of the infrastructure needed to conduct these trials, but also how we make some intermediate decisions on stopping certain arms and reallocating resources. Okay. One of the things that we also don't do enough, that we don't build into our trials, is really thorough interim monitoring. This is sometimes hard when we have few patients, but when we have a moderate number of patients in some rare diseases, especially when we have multi-centered groups, we can absolutely design in reasons to and set boundaries for early stopping, for futility monitoring, meaning that we see no signal early on, so we stop. For early efficacy monitoring, so we, again, stop early when we know the treatment is working better. This is generally done in randomized trials. We do see some of this work in more observational studies, too, when we're thinking about comparative effectiveness. But we also have to plan these kind of interim monitoring steps ahead of time. Sample size re-estimation is another kind of interim step that we could consider. So we know that when we design trials, we have preliminary evidence about those design effects. And they're often in different kinds of patients from different institutions, and we don't always see the same things that we expect. And so for sample size re-estimation, there are a number of different ways that we can think about doing this, but we essentially kind of use the data that we've accrued so far, some of it, not necessarily estimating the interim effect size, but looking at what we call nuisance parameters, the things associated with variation that we often have to guess at or make several guesses at when we're designing these trials. We can estimate that from our current trial, re-estimate sample size, and then move on, maybe increasing that trial by 10 or 20 patients, sometimes 50 patients, depending on the magnitude of the trial. And in the end, we kind of meet our design thresholds, and we don't come to the end of the trial saying, oh, we should have recruited 20 more patients. Maybe we would have met, the criteria for moving this drug or this therapy further. Again, this takes some upfront planning. We often have kind of advisors to do this outside of the study team, even outside of the study stat group. I think along the lines of some of the work Dr. Bird spoke about too, when we think about repeated measures designs, we're thinking about measuring patients over time, but this, I think, is really specific for the study outcomes, but also for samples that are taken on patients and banked over the course of trials. These are really powerful for later hypothesis generation, but also in terms of the key study endpoints. Repeating measurements on the same patient very much utilizes all of the resources that you're collecting. We might see mentioned designs called N of 1 trials. These are the most extreme case of the repeated measures design, where you recruit one patient and measure them many, many times. We don't generally recommend this. I think this makes a lot of assumptions, but in the most rare cases, I suppose this could provide some information. Crossover designs are another thing that we could consider. These are designs where patients under certain treatments can be given one therapy, and then there's a period where they're given no therapy and then given the second therapy. And we can then utilize the same patient to look at both therapies. Of course, we make some assumptions in all of these designs, but these designs carry fairly strong assumptions that we don't have carryover effects, meaning that the patient and the washout period is fairly short. The carryover period of the drug effects is fairly short, and that we hope that the disease is stable through time. We could also think about, all of these designs I've just spoken about are referring to designs where we're hopefully randomizing patients. We can, of course, use historical data, and this step often happens first before the randomized trials. We could use data from historical controls from the same university, the same institution. We can think about pre- and post-designs. This is also kind of another iteration of a repeated measures design where we just have one group, and we're looking at outcomes or disease status pre- and post. I think also when we have limited resources, we have to strongly consider, outcomes that we're measuring, ease of measurement, patient burden, and then this compromise between definitive and surrogate endpoints. The surrogate endpoint could happen sooner. It often happens sooner, but it could provide some very good information for us in terms of decision-making for patients. And I think this is just a summary. We have lots of things to consider. Some of these aspects we can take into account together. We can use, interim monitoring and sample size re-estimation together. We can think about how those correspond to the overall study design. So it's not just one aspect that we can use. We can bring all of these pieces to make the most efficient use of the patient data that we will be collecting. Let's see. I think there might be, oh, no, there aren't any more slides. Okay, so I think we have some other things to discuss. Slides. Are there more slides? Mm-hmm.
So while we're getting ready,
Sure.
Can I ask, because, so we'll pretend I'm a clinician, which I am. Dr. Ardwin and I study autoimmune disease, and our patient populations are very tiny. And so it's really very difficult to generate the sort of ends that power a cardiovascular trial or an oncology trial for some of the larger. So you had spoken, and I'm going to throw out just a couple things. One of the things that we're doing in nephrology now and is called it's called a remission clinic. And the design is that you intervene using known therapies and push them to the maximum. And so now I'm a clinician talking to you to design the trial, but there is no control group. So the control group that's been used are people who've had some of these interventions in a randomized controlled trial. So in other words, they would be in the placebo arm of a randomized controlled trial. So you have very good phenotyping and clinical follow-up throughout an entire course. as a statistician, what do you think about that as a historical control rather than simply, oh, I've got 10 people in my lab that, or in my cohort that I've followed in the past, and that's what I'm going to use?
I think it often depends on the kinds of information you can get from those external cohorts. So if you have more than summary data from those trials, if you have potentially de-identified individual level data, that may be more powerful than the 10 patients you would have from your own institution. I think in terms of levels of evidence, of course, that's not going to be our top tier, but when we have very rare diseases, we often have to make exceptions. So it would be probably better than the 10 patients you'd get from.
So you can see that you could do a similar thing in, for example, autoimmune diseases, where now there's been many, many trials of patients, for example, with lupus, who we've treated with standard of care therapies.
Right.
And if we wanted to test a new therapy against standard of care, we could potentially maximize our recruitment to just the patients getting that therapy and compare them to the standard of care groups from the past. The question is, and I think for the audience, is can you actually get these data at the patient level? And it turns out now that in a lot of these drug company-sponsored trials, they are starting to make the data public. And so you can get them off the database. They're all de-identified, but you have the clinical characteristics, the treatment, and the outcome. So I think this is a doable study design that might actually facilitate in some of these very difficult to recruit for populations.
Sure. I mean, I think we have, definitely over the past 10 or 15 years developed lots of more robust and rigorous techniques for causal inference outside of the randomized trial setting. And so those work better when we have more patient data. So when we have larger databases, when we only have 10 or 20 or even 50 patients, it's still quite limited in terms of what we can still make inference to and about. But I think the resources from these drug companies on patients with some of these rare conditions, even if it's not what we'd prefer, is what we have. And we can, I think we have some very good results in terms of methodologic results, in terms of how those methods perform in the ideal setting, and they do quite well if we can make some not terribly strong assumptions. So yeah, I think we can move forward with some of those that way.
So if you're going to do that, would you do like a age and sex match, like case control kind of a study, or would you just take all the control group in the study that is statistically less dramatic?
I think how we'd often think about it is that we'd think again as if we were moving prospectively through a trial. So we would take from wherever we could have patient-level data, set an inclusion-exclusion criteria, take all of the patient data that we can get, and then we don't have to select based on the outcome. We really want to pretend like we're seeing this prospectively from the beginning. So we think about who gets what treatment, and we try to balance treated and non-treated or whatever treatment groups, however we define them, as best we can. And we have weighting techniques, we have matching techniques, we have stratification techniques. We can use all of these to balance patients who are in different treatment groups. And then after that, we can look at their outcomes. And like a trial, we wouldn't look at the outcomes until we were satisfied until the patient groups were adequately matched. So that's in the best case.
And by using existing data from previous clinical trials, I think it would make the job easier of matching, because you tend to then have inclusion that would be sort of random, right, at the start of those trial, and for the disease you're looking at, it would include the patient population that you want to target with your new therapy, because you choose those particular trials.
Yeah.
So I was just going to tell a little bit of a story about, I'm a pediatric rheumatologist, and so pretty much every disease I treat is a rare disease with a couple of exceptions. Everything meets that prevalence rule of 1 in 200,000. It's a real challenge for our community. And I'm just going to talk about how that experience has kind of shaped some of the trial design that we've done. I'll try to go through these pretty quickly. So some of the challenges we experience in pediatrics on top of the challenges of rare disease are listed on this slide. And I'll just point out a couple of things. One is some of the parents are pretty skeptical about research, which can be a big challenge. They're willing to accept that risk for themselves, but they more often have reservations about a child. Children are not small adults, and so making inferences from adult data can be very dangerous because of differences in metabolism experience. And in pediatrics in general, and particularly in pediatric rheumatology, we have a dramatic shortage of subspecialists, which makes it really challenging to have the manpower to recruit patients and do the research. And just to demonstrate, just for pediatric rheumatology, all the states in white here have 4 or more pediatric rheumatologists, which is considered the highest bar. And the purple have 0. So we really have a small group of people to perform this work. But some of the opportunities are, of course, with drug development. The FDA has given several incentives recently for drug development. Children are ideal to study in many ways because they have fewer comorbidities and exposures compared to adult counterparts. And especially in pediatric rheumatology, we've really had a burning platform for change, really wanting to bring evidence basis to what we're doing. So we have, in rheumatology, have you heard today about the power of networks, and pediatric rheumatology is no exception. We've established the Childhood Arthritis and Rheumatology Research Alliance, which is called CARA, which is really young network. It's only 14 years old, but we have over 95% of all the pediatric rheumatologists in North America and participating in this, which is a pretty amazing penetrance, I feel. And we've secured about $40 million in research funding to start 120 sites and over 400 investigators. And the power is in numbers. So JIA is one of the diseases, juvenile idiopathic arthritis, that is not considered rare. That comprises 73% of the patients in our registry. But you can see all the other are very rare. I study lupus, and that's the next biggest one in the teens percent. But some of the other diseases we take care of, like pediatric scleroderma, are just extraordinarily rare. And the question was, how do we go about learning anything about these populations of patients? So within the CARA portfolio, we do a lot of different types of research, but the one thing that I'm just going to focus on here is some of the interesting work we've done with comparative effectiveness research. And this is a busy slide. I won't spend too much time on it, but what it tells us is that through this network, we've been able to grow in terms of numbers and sort of our research portfolio, and we have performed several randomized controlled trials, but we recognize the significant cost of these, the difficulty recruiting patients, and really wanted to focus on how could we possibly do more practical clinical research, especially for these rare diseases where it's virtually impossible to secure funding. So this has led to the development of what we call consensus treatment plans. And we as an organization have agreed to develop these sort of consensus on how we will treat patients so that we can actually reduce variation and learn more about how well our treatments are working. So just to give you one example, one very rare disease is localized scleroderma in children. And we surveyed pediatric rheumatologists and said, how do you treat localized scleroderma? And this is what we got, this busy pie chart. How on earth do you study that to see what works? So we developed these consensus treatment plans, which are really done according to very rigorous criteria where there's a very in-depth literature review. We meet together, we do case-based surveys, we have consensus meetings, we do voting, we do more consensus meetings until we achieve over an 80% consensus on sort of very key aspects of how we're going to treat, monitor what the outcomes will be, what are we going to follow, how are we going to measure, how are we going to determine success. So with the example of localized scleroderma, we've been able to reduce that crazy pie chart. On the left-hand side, you see all the different corticosteroid regimens that people used, and on the right, the methotrexate, and we reduced it from a ridiculous number to basically 3 different corticosteroid regimens and 1 methotrexate regimen. So now we're in a position, yes, where we can study this and see if this works. And so this result has led to securing funding through NIAMS for a pretty robust trial comparing these 3 results, which is ongoing, which we're expecting results in the fall. And also been able along the way, as you were commenting on collecting biosamples, so serum and skin biopsies to allow us to understand the pathophysiology. And we've done this in other diseases like systemic JIA, pediatric lupus nephritis, et cetera. So it's just another model of how we can alter study design in the setting of a rare disease to learn more about our diseases.
So can I play off the consensus? one of the things we're learning, I also study lupus with Dr. Ardwin, but in adults. We're trying to work with the FDA to make new response criteria, which you've heard earlier is absolutely essential to designing a clinical trial. And I'm not going to bore you with the details of the response criteria. Suffice it to say that we're developing our hazard indices for long-term poor kidney outcomes with lupus nephritis. And the question has arisen as to what would be, if you reduce the risk, if a new therapy reduces the risk, for example, of chronic kidney disease, how much of a risk reduction is necessary for this to be clinically relevant? So, everyone sitting in the room can decide what they think it is, but of course, it really has to pass the muster of the FDA. Mm-hmm. So, I can tell you how we're doing it, but how would you all approach that? How do you form your consensus, or how would you decide on this sort of thing?
How do I decide?
Yeah.
I look to my clinical colleagues.
Okay. Well, and that may be just exactly right.
Well,
I don't usually decide. I mean, I think we usually know when that design or that change is way too small to be really relevant.
Statistically.
Well, or practically. I mean, when we think we only want to change by half a standard deviation unit and we need 3,000 patients, that's not feasible. But is that also clinically relevant to us? Maybe, if it's something very prevalent.
Mm-hmm.
Well, I mean, in these examples, the one I gave you about localized scleroderma, we actually had to create an outcome measure first, and then do trials to decide, sort of small pilot trials to decide what would be the clinically acceptable change. Yeah. So it's very challenging.
So the way we're doing it is very similar. There is no, as far as I can tell, statistical way to reach a consensus for a clinical outcome, so it has to be based on clinical experience. But the way we're handling it, which I think is effective, is we're working with the FDA to decide on what is a clinically relevant reduction in the rate of future chronic kidney disease for approval of a therapy. If it 5%, 10%, 25%, and the idea is to take clinicians and the FDA and put them in sort of a room to come up with this. And that way, then you set the standard for how you do your trials down the road. And I think, while it may not be perfect and you can't prove it statistically, clinically you know what's important to your patient population, and this is a way to handle that particular issue.
One question I had for you is, one of the things that I'm really struck by is the fact that in some of these analyses, especially in trial studies, there's very little sensitivity analyses that are done. So in terms of sort of measuring the variance around the effect size, you would think that some sort of simulation exercise would be useful, to sort of counteract some of the problems with small sample sizes, those sort of issues. What do you think about more of that rigor added to it?
In a randomized trial design, these sorts of trial results. So I think it would depend. So if a randomized trial is conducted well and we can capture the endpoints and there isn't a lot of, there's compliance as best we know, the trials are analyzed per intention to treat. So analyzed as they were randomized, which is not basically on the treatment that they may have received or wanted to get. This provides us an unbiased estimate of the treatment effect when we have a sufficient number of patients measured on that outcome. In the case where we have lots of missing data, it's a problem. So I think we often do sensitivity analyses for randomized trials, especially when there's a lot of dropout, a lot of crossover. Of course, that's part of the clinical trial conduct, but there is a whole lovely FDA document on missing data and how to prevent it and how to ameliorate the effects of it. And we have, various statistical techniques. It's to look at the impact of missing data. We could spend probably a whole day talking about missing data, but so I think that's maybe one step. We often include some kind of sensitivity analysis, but again, in publication, sometimes that doesn't translate well, or it gets put in supplemental material.
Quickly, do we have any other questions we want to ask the study design panel before we move on? And we are getting a little into the next session, but I want to make sure that we have an opportunity to ask, because I think this is a sort of an important topic. So if we have any others? Sure.
You talk about the number of patients, so what do you consider as reasonable number?
A reasonable number?
Yeah, you mentioned 10, 20, I mean, exactly.
Oh, I think it depends. I mean, I think it depends on what we're measuring and the kind of phase of research that we're in. I mean, we do phase 1 trials all the time with 6 or 9 or 12 patients, but of course, that's very preliminary evidence. When we're thinking about moving forward in terms of looking at efficacy, I think it really depends on what's clinically relevant and then what's too few per the design consideration. So sometimes we see a very big impact with one treatment with just 20 or 30 patients, and we can have a really definitive answer about that therapy, but that's not often the case.
So in a lot of our trials in lupus and lupus nephritis, we start out with a sample size estimation based on known treatment effect size. And the reality is that we often don't meet that recruitment, or if we're trying to meet that recruitment, it takes several years and the trial sort of fades away. I know we're running out of time. I'll leave you with one last point, and that is we are often dealing with diseases that have very non-homogeneous presentations. And one of the things we've been advocating is that if we can sort of know ahead of time our population and their genetic and clinical phenotypes, we can reduce the sample size by having a much more homogeneous population. And that's what we're pushing for now as we help pharma design some of these drug trials, is to try to get the patient population as homogeneous as possible. And also based on the pathophysiology of the disease and what the drug is intended to do, maybe include those patients, but not necessarily patients that might be having a different pathogenesis of the same disease, because there's often different pathways for the same disease phenotype.